Hello everyone, I hope you're all doing okay. This is our first video on the basal ganglia anatomy. To contextualize the topic a little bit better, we'll be talking about the location of the basal nuclei, both from a structural as well as from a functional perspective, and the overall bigger picture of the motor system organization. Next, we'll talk about the 3D gross anatomy of the basal nuclei or the basal ganglia. Then we'll try to visualize the basal nuclei on the horizontal as well as on the coronal sections taken through the brain at locations where the basal nuclei are situated. So before we start describing the basal nuclei, how about if we do a quick recap of the motor system organization using a schematic which we used in one of our earlier videos on the motor system. Now, you would have seen this schematic already if you had listened to my first video on the motor system. Here in the center, you can see the horizontal sections taken through different levels of the central nervous system. You can see the horizontal section taken through the spinal cord over here. This is a horizontal slice taken through the lower medulla. Then this is the horizontal section of the upper medulla oblongata. You can see the section of the pons over here, followed by the midbrain. And then this is the level where the internal capsule and the thalamus are situated. And lastly, you're looking at a coronal or a side-to-side slice which has been taken through the cerebral cortex. I've already described these sections in a little bit more detail as part of my videos on the ascending and the descending motor pathways. Now, in the original video, we discussed that the reflex arc is the basic foundation or the building stone of the motor system. And we explained that the reflex arc basically comprises of 5 different components. There is a receptor which senses the sensation from the external environment and sends this information through the afferent sensory neuron into the central integration center, which could be the spinal cord or the cerebral cortex. The integration center then analyzes that information and sends the motor command through an efferent or a motor neuron to an effector organ, for instance, a muscle or a gland. Then we said that the reflex arc was under the influence of the higher centers, such as the cerebral cortex over here, which was influencing the reflex arc through the descending neural pathways shown by the red dotted line and by the pink dotted line over here. These descending neural pathways, these are actually the upper motor neurons. Some of them are excitatory, whereas some of them are inhibitory on the reflex arc. However, the dominant effect is that of inhibition, and that is why when somebody gets an upper motor neuron lesion at any level inside the central nervous system, then that actually results in the loss of that inhibition on the reflex arc, making the reflex arc hyper-excitable. And that is why in an upper motor neuron lesion, we usually see spastic paralysis, meaning thereby then when we do a neurological examination, we'll see the weakness, which obviously would be there because of the upper motor neuron lesion. However, the weakness would be accompanied by hypertonia and hyperreflexia. This is contrary to the lower motor neuron lesion, where the weakness is accompanied by hypotonia and hyporeflexia. In other words, a flaccid paralysis. As part of the descending upper motor neuron pathways, we studied in detail the corticospinal tract, which extended from the cerebral cortex, shown by the red dotted line over here, going all the way down here to the anterior horn cells of the spinal cord. And then and in subsequent lectures, we will be talking about the descending upper motor neuron pathways over here, which are the corticonuclear pathways, shown by the pink dotted line over here, extending from the cortex all the way down to the cranial nerve nuclei. The corticonuclear tracts will influence the reflex arc at higher levels inside the central nervous system, such as the reflex arcs situated inside the brainstem. Then we said that there are other components of the motor system as well, which you can see in this schematic. These include the cerebellum and the basal ganglia. You can see that the cerebellum and the basal ganglia are connected with the cerebral cortex with the help of various neuronal loops. However, what you can see over here is that they are not having any direct access to the reflex arc, and so therefore they can modulate the activity of the reflex arc indirectly through modulating the function of the cerebral cortex. And that is why when somebody gets a lesion inside the cerebellum or inside the basal ganglia, then there obviously is some kind of a motor impairment, but we don't see an outright paralysis. Instead, we see some other problems with the motor activity, especially pertaining to the coordination of the motor activity. Now, although this video deals with the anatomy of the basal nuclei, however, we will just briefly highlight some of the important functions of the basal nuclei first before we jump on to the anatomical details of the basal nuclei. Although this video discusses in detail the anatomy of the basal nuclei or the basal ganglia, however, let's just highlight some of the important functions of the basal nuclei first before we delve deep into the anatomy of the basal ganglia. Right, so the basal nuclei play an important role in the initiation and in the execution of the voluntary movements, especially those which have been learned as a result of a lot of regular practice. For instance, when you learn riding a bicycle, for instance, or when you learn to drive a car, in the beginning, the activity is really novel to you, and so your cerebral cortex is heavily involved in that novel activity. You have to pay a lot of attention as to when you should be pressing the accelerator, when you should be pressing the brake, when at what time would you be changing the gears. However, once the activity has been done a number of times, then it becomes kind of second nature to you, and that motor plan for that motor activity gets transferred down to the subcortical area where the basal nuclei are situated, and the basal ganglia then play an important role in the initiation and execution of that motor plan which has been learned with experience now. Similarly, when you're climbing up the stairs, for instance, right, you know from your previous experience now that every staircase, every step in the staircase is going to be of a certain height because you've done that a lot of times before as well. So basal ganglia can actually predict and help calculate the length of the step which you need to take in order to climb up the stairs, and you don't even have to look at the stairs at that time. This is a voluntary activity which has been initiated by the basal nuclei, and an important aspect of the voluntary activity is that they basically are calculated based on predictive function, based on something which you've done so many times in the past. However, once the activity becomes novel, then the cerebral cortex has to step in, and the basal ganglia kind of becomes less involved in that kind of an activity. This is the reason whenever there is any lesion inside the basal nuclei, the voluntary activity gets impaired. However, we don't see any outright paralysis in that patient. Instead, we see some other kinds of problems with the motor voluntary activity, especially the problems associated with the coordination. And that is why in diseases such as in Parkinson's disease, which affect the basal nuclei, we see motor problems such as tremors, which are associated with the lack of coordination of the muscle activity, or we could actually see the motor activity being slowed down. We see a lot of stiffness and rigidity in the body of the Parkinson's disease patients. The motor activity gets slowed down, which is known as hypokinesia. One thing which is important to note over here is that since the predictive voluntary activity gets impaired, the reflex activity is still intact. For instance, if you ask a Parkinson's disease patient who has a lesion inside the basal nuclei, if you ask that patient to pick up a glass of water on the table, then he might not be able to execute that activity smoothly. You might actually see a lot of shaking in his hands. His activity might be really slow in terms of reaching to the glass and holding that glass of water. However, if you suddenly throw some object on the Parkinson's disease patient, such as a ball, for instance, and he might be able to reflexly respond, outstretching his arm. So basically, the point which I'm trying to make over here is that the reflex actions are still intact. However, the voluntary predictive functions get impaired in the basal nuclear lesions. Then the basal nuclei are also linked with controlling the eye movements, especially the fast conjugate eye movements, which are known as the saccades. That is why in people with Parkinson's disease, the saccadic eye movements, they get impaired as well. And what we typically see in Parkinson's disease is that they've got jerky saccadic eye movements, which are known as the step saccades. And that is also one of the reasons why the eye movements, the saccades, have been used extensively in Parkinson's disease patients to be used as a prototype to study the motor impairment as a whole in the disease. Then the basal nuclei have got extensive connections with the prefrontal cortex as well, and that's why a cognitive decline is an important clinical manifestation as well once the Parkinson's disease has spread extensively inside the brain. Then the basal nuclear circuitry is extensively linked with the limbic system circuitry as well, and the limbic system is linked with emotions, and that's why we also see a lot of emotional or the mood elements in a Parkinson's disease patient as well. Now, before we begin talking about the basal nuclei, let's just find out which parts of the brain are collectively called as the basal ganglia or the basal nuclei. Actually, before we do that, I just want to clarify some definitions over here. So a nucleus by definition is an accumulation of neural cell bodies inside the central nervous system. On the contrary, a ganglia by definition is an accumulation of neural cell bodies outside the central nervous system, that is inside the peripheral nervous system. However, in the literature, you'll come across the terms basal ganglia and basal nuclei being used interchangeably. So don't get confused by these terminologies, please. They both refer to the same structure, which is the subcortical basal nuclei. Right, so the basal ganglia or the basal nuclei are large subcortical structures which consist of various interconnected nuclei present inside the forebrain, inside the diencephalon, and in the midbrain. These include the striatum, the globus pallidus, the substantia nigra, and the subthalamic nucleus. Now, in order to understand the location of the basal nuclei, we need to first understand the ventricular anatomy, because the ventricles are centrally situated structure within the central nervous system, and the basal nuclei are situated around the ventricles. Therefore, in order to understand the location of the basal nuclei, we first need to understand where the ventricles are situated within the central nervous system. Now, inside each part of our central nervous system, there is a fluid-filled space situated, which is known as the ventricle. The spaces have been shown color-coded in blue over here, while the brain and the spinal cord actually have been shown in the background. So the ventricles have been overlaid on the brain and the spinal cord. But in reality, the ventricles are situated within the brain and spinal cord. This is just to help you understand and to contextualize the location of the ventricles a little bit better. The fluid present inside the ventricular system is going to be the cerebrospinal fluid. And this entire system of interconnected spaces, this is known as the ventricular system. These spaces include the lateral ventricle. This lateral ventricle, which is a large C-shaped structure, this is present within each cerebral hemisphere. So there would be two lateral ventricles, one in each cerebral hemisphere, which are interconnected with each other. And we'll talk about the connection in a little bit. You can see that the lateral ventricle has got a frontal horn over here, which is inside the frontal lobe of the cerebral cortex. Then we've got the body of the lateral ventricle over here, which is more or less residing inside the parietal lobe. And we've got the occipital horn or the posterior horn, which is inside the occipital lobe of the cerebral cortex. Then down below over here, we go into the inferior or the temporal horn inside the temporal lobe of the cerebral cortex. Then we've got the third ventricle over here, which is basically a midline space. And so that is why when we look at the prosections and we take a midline sagittal section of the brain to have a look at the medial surface of the brain, we'll be basically cutting through the third ventricle, and we'll be looking at the structures forming the lateral wall of the third ventricle. So the structures which we're going to see in the midline sagittal section would be the thalamus and the hypothalamus. Now the third ventricle is connected then with the lateral ventricle through an interventricular foramen of Monro, which is basically a channel which allows the fluid, which is the cerebrospinal fluid, to flow from the lateral ventricle into the third ventricle. So now you can imagine that there would be one lateral ventricle in each of the cerebral hemisphere containing the cerebrospinal fluid, and the CSF from each of the lateral ventricle would be flowing through the interventricular foramen of Monro, one on either side, into the third ventricle. So there would be two interventricular foramen of Monro's as well, and they would be draining the CSF into the one midline third ventricle in the center over here. Now down below over here, we've got the region which is known as the brainstem. Here we've got a space over here which is called the fourth ventricle. The fourth ventricle is bounded posteriorly by the cerebellum and anteriorly by the pons and the medulla oblongata mainly. So this is another extension of the ventricular system which is present inside the brainstem region now. Then we've got another part of the ventricular system here, which is called the cerebral aqueduct. You can see part of the cerebral aqueduct hiding behind the C-shaped lateral ventricle, but the cerebral aqueduct is a continuous structure or a channel which connects the third ventricle with the fourth ventricle, allowing the CSF to pour down from the third ventricle into the fourth ventricle over here. The cerebral aqueduct is that part of the ventricular system which is present inside the midbrain. Then from here down, the fourth ventricle then continues down as the spinal canal, which is basically an extension of the fourth ventricle inside the spinal cord. So you can appreciate over here that there is a space within every part of the central nervous system, and this interconnected system of space, this is known as the ventricular system. The fluid present inside these spaces is the cerebrospinal fluid, which is present within the central nervous system. From these spaces, the fluid is then going to pour out through certain holes in the ventricular system to come outside the brain inside the subarachnoid space, and we'll talk about that sometime later. However, the main motive of explaining the ventricular anatomy briefly over here is to help you understand the basal nuclei relationships when we talk about them in a bit. Right, so now here you're basically looking at a midline sagittal section which has been taken through the head and neck region. You can see the floor of the cranial cavity over here. So the brain is residing inside the cranial cavity. You can see different aspects or different components of the ventricular system over here. So for instance, if you look over here, see this is the lateral ventricle. This is the body of the lateral ventricle, and you can see that it is extending anteriorly. It goes anteriorly into the frontal lobe as its frontal horn, and then it's going to extend backward into the occipital lobe as the occipital horn, which we cannot really see clearly over here, but at least the body of the lateral ventricle is clearly visible, and the floor of the body of the lateral ventricle is clearly visible over here as well. Then if you look over here, you can actually see the region where the interventricular foramen of Monro is going to be present. This is the area where the third ventricle is going to be situated, and the third ventricle is going to be connected with the lateral ventricle through the interventricular foramen of Monro. Now if you jump over to this prosection over here, this region is a little bit more clear over here. Once again, you're looking at a midline sagittal section here. This is the anterior aspect, this is the posterior aspect of this prosection. You can see the body of lateral ventricle here very clearly, and see the structure over here. These are some white matter tract fibers which are known as the fornix. We'll talk about those sometime later. This egg-like structure over here at the back is the thalamus, and this space over here between the thalamus behind it and the fornix in the front and at the top, this is the interventricular foramen of Monro. And you can imagine now that the fluid, which is the CSF, is going to flow from the lateral ventricle through the interventricular foramen of Monro and then coming into this region over here. This is a region which is the third ventricle, and it is bounded by the thalamus laterally and by this triangular region over here called the hypothalamus laterally. So the thalamus and the hypothalamus are the lateral relations of the third ventricle. We're actually inside the cavity of the lateral of the third ventricle here. From here, you can actually extend down into this region, into this channel over here, which is called the cerebral aqueduct. This is the extension of the lateral ventricle inside the midbrain. So this region here belongs to the midbrain. So we're looking at the brain stem now. It's kind of more clear over here. Probably this is the brain stem over here. Well, the midbrain is not really that clear, but you can imagine the cerebral aqueduct being over here. It continues down into this space over here, this diamond-shaped space. This is the fourth ventricle, and you can see this is bounded posteriorly by the cerebellum and anteriorly by this bumpy region, which is the pons and the medulla oblongata down below here. The fourth ventricle then continues into the spinal cord as the spinal canal, which is obviously within the spinal cord, so you can't really see that on the prosection. Now this is once again a medial view of the cerebral hemisphere, showing a midline sagittal section of the brain. This is the left cerebral hemisphere, so the occipital lobe is at the back, the frontal horn or the frontal lobe is going to be in the front over here. This is the area where the lateral ventricle is going to be situated. It's bounded over here. It's covered over here by this sheet, which is known as the septum pellucidum, which is actually the medial wall of the lateral ventricle. And if I actually cut through it, I'm going to go into the body of the lateral ventricle. Once again, I can see the cerebral, I can see the thalamus over here and the hypothalamic region over here. So this is the area where the third ventricle is situated. And then we go down through the cerebral aqueduct into the fourth ventricle. And so fourth ventricle can be seen clearly bonded by cerebellum at the back and the pons and the medulla in the front. Fourth ventricle then continues down into the spinal cord as the spinal canal. Once again, not clearly shown over here. Now that we have developed a basic understanding of the 3D orientation of the ventricular system, let's begin by identifying the basal nuclei, which are situated around the ventricles. Let's begin with the striatum, which comprises of two main subparts, the caudate nucleus and the putamen. The caudate nucleus is a C-shaped structure which has got a head, a body, and a tail to it. Now I'm going to use this little video to help you understand the 3D anatomy of the ventricles, the thalamus, and the relationship of the basal nuclei, such as the caudate nucleus, to these structures. So let's just begin with the 3D anatomy of the ventricular system once again. What you can see over here is the C-shaped lateral ventricle. See, this is the frontal horn, the inferior horn, then the occipital horn at the back, and then you can see the third ventricle in the center over here. This third ventricle is connected with the fourth ventricle with the help of this channel, which is called the cerebral aqueduct, which we discussed already. Then what you can see over here is that we've got a nucleus which is called as the thalamus, which we are going to place just lateral to the third ventricle. So you can see thalamus is the lateral relation forming the lateral wall of the third ventricle. You can see the two thalami on either side of the third ventricle, which is a midline structure. And you can also see third ventricle connected with the lateral ventricle through the intraventricular foramen of Monro, one on either side. Now we've got the caudate nucleus, which is a C-shaped structure having a head, body, and tail to it. See, we're going to convert this into a C-shaped structure and then fit it into the large C of the lateral ventricle. So what you can see here is the caudate nucleus having a big head to it. The head of the caudate nucleus is actually the inferior relation of the frontal horn of the lateral ventricle. The body of the caudate nucleus over here is then going to be forming is then going to be related to the body of the lateral ventricle over here, forming the floor of the body of the lateral ventricle, and then the tail over here then forms the roof of the inferior or the temporal horn of the lateral ventricle. So you can see the 3D orientation of the caudate nucleus, which is fitting into the large C of the lateral ventricle. Right, so now our next job is to find out how the internal capsule and the corona radiata, these are related to the basal nuclei. Right, so we already talked about the orientation, the location of the caudate nucleus, and we said it was a C-shaped nucleus fitting into the large C of the lateral ventricle. We can see the head of the caudate nucleus, the body, and the tail of the caudate nucleus over here. Now we're going to put in the corona radiata and the internal capsule fibers over here. So what are corona radiata and internal capsule? These are basically vertically oriented white matter tract fibers, and if I just put them over here into context, then you can clearly see that the corona radiata over here, these fibers are converging down over here to form this condensation of the fibers, which is known as the internal capsule. The internal capsule, from the internal capsule, these descending white matter tract fibers, they're going to pass through the peduncles of the midbrain, and you can clearly see over here that the internal capsule and the corona radiata is situated lateral to the caudate nucleus, to at least the major bulk of the caudate nucleus. And so therefore, if in a prosection we see the corona radiata and the internal capsule intact, then we would not be able to see the thalamus and the caudate nucleus. Both the thalamus and caudate nucleus, as well as the ventricles over here, the body of the lateral ventricle over here, and the frontal horn, these are actually the medial relations of the corona radiata and the internal capsule. And you can see that clearly over here as well. See the corona radiata internal capsule situated lateral to the caudate nucleus and to the body of the lateral ventricle over here. This is another frontal view of the same structures, and you can clearly see the corona radiata and the internal capsule situated lateral to the caudate nucleus. You're looking at the head of the caudate nucleus over here. This is the frontal horn of the lateral ventricle, and you can see the thalamus over here. So both the caudate nucleus and the thalamus are the medial relations of the internal capsule. Then we've got another nucleus which is situated lateral to the internal capsule, and that is called as the lentiform nucleus. The lentiform nucleus is a part of the basal nuclear system, which is comprising of two subnuclei. Those are the globus pallidus and the putamen, and we're going to visualize them in a bit. The globus pallidus in itself is comprised of two subparts, an internal segment and an external segment, and we call those segments as the globus pallidus internum and globus pallidus externum. Right, so let's go back to our video over here. We've seen this relationship already, where we saw that the corona radiata and the internal capsule were actually going down over here through the peduncles of the midbrain over here. The internal capsule, we said, was the lateral relation of the caudate nucleus and the thalamus, and obviously the ventricles are midline situated structures as well. Now we're going to find out where the lentiform nucleus is situated. So if you look over here, you're looking at the corona radiata and internal capsule from its lateral aspect, and you can see the caudate nucleus and thalamus are hiding behind it or medial to it, and this is the place where the lentiform nucleus is going to be situated. So first we're going to put the globus pallidus over here, and we said the globus pallidus has two subparts to it. The globus pallidus internum, which is going to be the immediate lateral relation of internal capsule, then globus pallidus externum, internum, and then lateral to the globus pallidus externum would be the putamen. Together, these three subparts actually form the lentiform nucleus, and you can see now the lentiform nucleus being the lateral relation of the internal capsule, both the globus pallidus as well as the putamen, and you can see the quadrate nucleus and the thalamus being the medial relation of the internal capsule. Internal capsule is therefore sandwiched between these different subnuclei of the basal ganglia. Now, as part of the basal nuclear system, I'll talk about a specific region which is known as the nucleus accumbens. And then I'll talk about some cellular bridges which connect the caudate nucleus with the putamen to form a functional and structural entity which is known as the striatum. Now see anteriorly over here where the putamen fuses with the head of the caudate nucleus, this region over here which is being highlighted here, this is known as the nucleus accumbens, and this is actually where the neural circuitry of the voluntary basal ganglia activity gets hooked up with the emotional circuitry of the limbic system. So you can well imagine that how this interconnectivity basically allows the emotions to pour into the voluntary motor actions. Then we've got some cellular bridges over here which are connecting the putamen on the outside with the caudate nucleus on the inner aspect of the internal capsule. So these cellular bridges are basically gray matter which are passing through the internal capsule and connecting the two nuclei with each other, consequently forming one structural and functional entity which is known as the striatum. So striatum basically comprises of the putamen and the caudate nucleus connected with each other through the cellular bridges. The striatum serves as a receiver end of the basal nucleus. So all the afferent neurons which are coming into the basal ganglia, they're going to be entering into the striatum part of the basal nuclei. Now I just want to clarify one thing over here. You might actually come across the term striatum and corpus striatum when you read the literature. The corpus striatum is technically a little bit different from the striatum. The striatum comprises of the putamen and the caudate nucleus, whereas the corpus striatum comprises of the lentiform nucleus and the caudate nucleus. In other words, what I'm saying is that the corpus striatum comprises of the putamen, globus pallidus, and the caudate nucleus. The globus pallidus is missing in the striatum. Striatum only comprises of two nuclei, the putamen and the caudate nucleus. Then the other components of the basal nuclei include the substantia nigra, which sends neuronal projections to the striatum and hence functionally and structurally connected with the basal nuclei and therefore considered part of the basal ganglia system. Now, to understand where the substantia nigra is situated, let's have a look at a horizontal slice taken through the midbrain. We have already gone over these sections when we talked about the ascending and the descending pathways, so we can do a quick recap of the horizontal slice of the midbrain over here, and then we'll find out where the substantia nigra is situated. So here, this is the horizontal slice of the midbrain. You can see the ventral aspect of the midbrain over here characterized by these two rabbit ear-like structures. These are the peduncles of the midbrain. Posteriorly over here, we can see these lobulated structures over here. These are the colliculi. So this region of the midbrain, this is known as the tectum. We can see this little part of the ventricular system over here called the cerebral aqueduct. Any part of the midbrain which is behind the aqueduct is known as the tectum. That contains the colliculi. The part of the midbrain which extends from the aqueduct all the way over here to this region here, this is known as the substantia nigra. This region is the tegmentum. So we've got the substantia nigra over here, which is forming the boundary of the tegmentum, separating the tegmentum from the cerebral peduncles. The substantia nigra can be seen in this prosection over here as well, which is basically a Mulligan stained section taken at the level of the midbrain. You can see the two peduncles of the midbrain once again very clearly over here, and you can see the aqueduct at the back over here, and you can see the cerebellum which is forming the posterior relation of the midbrain over here. This dark region over here which contains a lot of melanin pigmentation, that is the reason why this area is dark. This is known as the substantia nigra. The substantia nigra can then be subdivided into two subcomponents, anterior and posterior. The anterior part is known as substantia nigra pars compacta, and that is the one which sends dopaminergic projections to the striatum, and this is where the neurodegeneration starts in case of Parkinson's disease. The posterior part of that substantia nigra is known as substantia nigra pars reticularis, which is also part of the basal nuclear system, but the dopaminergic projections, they pop out from the substantia nigra pars compacta, which is the ventral part of substantia nigra. Now let's do a quick recap of the ventricles and the basal nuclear anatomy by looking at some of the images of various prosections of the brain. So starting from this prosection over here, this is basically a horizontal slice which has been taken through the cerebral cortex at a level where we can see the two ventricular cavities, the two lateral ventricles very clearly. You can see the body of the lateral ventricle over here. This is the frontal horn, this is the occipital or the posterior horn, and you can see the two lateral ventricles across the midline, one in each of the cerebral hemisphere. The inferior or the temporal horn is not visible over here because this is a section taken at a slightly higher level where you cannot see the temporal lobes. You can appreciate very nicely the floor of the body of the lateral ventricle here. The two main important structures which form the floor of the body of lateral ventricle are this structure over here, which is known as the thalamus, this egg-shaped nucleus over here. That's the thalamus, and we know the thalamus is just situated lateral to the third ventricle, inner to the internal capsule, and then over here this is the head of the caudate nucleus, which then continues behind as the body of the caudate nucleus. The remaining part, which is the tail, is going to be slightly situated at a slightly lower level because the caudate nucleus is a C-shaped structure, and we have taken a section at a higher level where the body and the head of the caudate nucleus are situated. This tuft of vessels, which you can see within the cavity of the lateral ventricle, this is basically choroid plexus. It is present inside the ventricles, and this is the region where the CSF is going to be produced. Now, if you look over here, this is now a coronal section, a side-to-side section taken through the cerebrum. So we can now see the lateral ventricles from the back over here. See, this is the midline structure called the septum pellucidum, which is separating the two lateral ventricles from each other, and septum pellucidum is going to be situated somewhere over here, but here we're looking at it in a more vertical fashion because we've taken a side-to-side coronal slice through the brain. You can see the body of the lateral ventricle continuing anteriorly as the frontal horn. We're looking at the lateral ventricle on either side from the back over here, and here this is the floor of the body of the lateral ventricle formed by the two structures called the thalamus medially and the body of the caudate nucleus laterally. See, we saw the thalamus and the caudate nucleus over here, the thalamus being here and the caudate nucleus over here. Now we're looking at a coronal side-to-side slice, and we're looking at those structures from the back over here. And you can probably now appreciate the C of the caudate nucleus. The caudate nucleus is going to continue down over here to form the C-shaped caudate nucleus. Then if you look at the lateral ventricle from the lateral side over here you can probably appreciate the C of the lateral ventricle much more clearly. You can see the occipital horn of the lateral ventricle over here, and then you can see the temporal or the inferior horn continuing down into the temporal lobe. The body is hiding over here, continuing as the frontal horn, but they're not clearly visible because they're hidden behind this piece of cerebral tissue over here. Now here you're looking at another prosection of the brain. This is actually a sagittal section, but it has been taken slightly lateral to the midline. So you're looking at kind of a parasagittal section taken through the brain. You can see the fibers of the coronal radiator over here, which are converging down over here at this level where they all condense together to form what is known as the internal capsule. Internal capsule then continues down into the peduncle of the midbrain or into the crus cerebrum of the midbrain. Now this is the region where the internal capsule is situated. So just lateral to the internal capsule, we can see the lentiform nucleus situated right over here. The lentiform nucleus was removed from this prosection, so you can see internal capsule clearly, but now the lentiform nucleus has been put into its place, and you can see the lentiform nucleus is laterally situated to the internal capsule. We cannot see the caudate nucleus and the thalamus over here because we know that the internal capsule is the lateral relation of the thalamus and the caudate nucleus, so they're going to be hiding behind or medial to the internal capsule and to the coronal radiator. The putamen part of the lentiform nucleus, however, would be connected with the caudate nucleus on the inner side through cellular bridges, and that is why we said that the striatum is one functional structural entity because it is connected. Now let's try to identify the basal nuclei in a horizontal slice of the brain. So if we take a horizontal section at this level over here where the internal capsule and the basal nuclei are situated, then the horizontal slice is going to look somewhat like this. Now see, this is a horizontal section taken at a level where the internal capsule was situated. You can see in this illustration over here, this is the V-shaped internal capsule. The internal capsule has got a geno or an angle to it over here. This is the posterior limb of internal capsule, and this is the anterior limb of the internal capsule. All the coronal radiata fibers, they are converging in this region over here. The posterior limb of the internal capsule is related medially over here to the thalamus. So thalamus and the caudate nucleus in the front, they're both medial relations of the internal capsule. Now here, since we're looking at a horizontal slice, so we are only visualizing the head of the caudate nucleus, but actually the body of the caudate nucleus is then going to continue behind over here. It's a C-shaped structure, and it's going to continue behind over here and then going to go down as the tail of the caudate nucleus. But the level at which the slice has been taken, that only shows you the head of the caudate nucleus anteriorly. Then lateral to the internal capsule, you can see the lentiform nucleus, and we know the lentiform nucleus comprises of the putamen, globus pallidus externum, and then globus pallidus internum. So you can see the basal nuclei situated all around the internal capsule. Now this is actually a cross-sectional view of the same horizontal slice taken through the brain, and you can see the lateral ventricles over here very nicely. This is the posterior aspect, so we can see that these are the occipital horns of the lateral ventricle. These are the frontal horns of the lateral ventricle over here. You can appreciate the internal capsule, the V-shaped internal capsule over here. The V-shaped internal capsule is then related medially to this bumpy tissue mass over here. This is the thalamus at the back, and then you can see the cut section of the head of the caudate nucleus over here anteriorly, just like we talked about the head of the caudate nucleus here anteriorly, medial to the anterior limb of the internal capsule. Then lateral to this V-shaped internal capsule, we can see this dark tissue mass over here, this cone-shaped dark tissue mass. This is the lentiform nucleus comprising of putamen and globus pallidus. Now if we do a Mulligan stain on this section, then all the gray matter is going to appear blue in color. We can see that very nicely over here. The internal capsule, which is basically white matter tract fiber, they still appear white in the Mulligan stain, whereas the thalamus medially and the head of the caudate nucleus medially, these are gray tissue masses, so they are now appearing blue in color. You can see the frontal horns of the lateral ventricle very nicely over here. Lateral to the internal capsule, we can appreciate the cone-shaped lentiform nucleus, which is also kind of stained blue over here because this is gray matter. Now there are some other structures which you will be able to see more clearly on the prosections when you see them in real life, and those structures are as follows. These are the lateral relations of the lentiform nucleus. So just lateral to the lentiform nucleus over here, this whitish region here, this is basically known as the external capsule. So basically, if you follow these structures, you'll see that the structures are situated in such a fashion that we've got some gray matter followed by the white matter, and then the gray matter, and then white matter, and so on. So the most medial structure over here is the thalamus and the head of caudate nucleus, which is basically gray matter. Then lateral to those structures, we've got the white matter tract fibers, which comprise the internal capsule. Then lateral to internal capsule, we've got another tissue mass of gray matter, which is known as the lentiform nucleus. Then lateral to the lentiform nucleus, we've got again white matter tract fibers here. These are the external capsule. Then lateral to external capsule, we've got a gray matter tissue once again, and that gray matter tissue is known as the claustrum. It's not really clearly visible over here, but you can actually see this bluish line over here. That is the gray matter tissue, which is known as the claustrum. Lateral to the claustrum, then you can again see this whitish area over here. This is the extreme capsule. This is once again white matter. Then lateral to the extreme capsule, we've got this bluish region over here. This is the insular cortex, and insular cortex once again is the gray matter. Then if we go lateral to the insular cortex, then we're in a region over here which is basically the lateral sulcus. So we've gone outside the cerebrum now. Now let's have a look at a coronal section, or a side-to-side section taken through the brain, and let's try to identify the basal nuclei on the coronal slice once again. So see, we talked about the horizontal section of the brain already. Now I'm going to take a coronal side-to-side section at this level over here where the thalamus is situated, and let's see how the basal nuclei look on the coronal slide. So this is a coronal slice which is taken through the brain, and you can see that we're looking at the slice from the front over here. Down below over here you can see the optic nerve and the optic chiasma and optic tract, and then if you go a little bit higher up over here, we can see a midline structure here called the septum pellucidum, which is separating the two bodies of the lateral ventricles from each other. Lateral to the lateral ventricle over here we can see this whitish region here. This is a Mulligan stain section, so anything which is white matter appears white, anything which is gray matter is going to appear blue in color. So you can see the internal capsule over here. Medial to the internal capsule, this bumpy region over here, this is basically the thalamus, and thalamus is a gray matter tissue, that's why it's been stained blue over here. Lateral to the internal capsule, this bluish region over here, that represents the lentiform nucleus. So we follow the same rule over here as well. We've got structures which are gray matters and white matter, and they're present. So we're going to follow the same rule over here. We're going to see that different structures, different basal nuclear components are going to be present in such a way that you're going to find the gray matter followed by the white matter, then gray matter and white matter, and so on. So if you look over here, we've got the thalamus, which is gray matter, then we've got the internal capsule, and this is actually the posterior limb of internal capsule because we are looking at the thalamus, and thalamus is related medial to the posterior limb of internal capsule. Had this been the head of the caudate nucleus, then this would have been the anterior limb of the internal capsule. So we're looking at the posterior limb of internal capsule, which is white matter. Then lateral to it, we've got the lentiform nucleus, which is once again the gray matter. Then lateral to the lentiform nucleus, we've got this extreme capsule, we've got this external capsule, which is white matter once again. And lateral to this white matter, we can see a streak of bluish region over here. This is basically the claustrum, and claustrum we said was gray matter. There is no functional significance associated with the claustrum. Embryologically it is more of a vestigial organ, but it comes up in the spot, so you have to know where the claustrum is situated. Lateral to the claustrum, we've got again this whitish region over here. This is now the extreme capsule, which is white matter. Lateral to the extreme capsule, we've got once again this bluish region here. This is now the insular cortex, or the insula, which is once again gray matter. And then once we go outside the insular cortex, then this region over here, this is where the lateral sulcus is situated. So we're outside the cerebral cortex now, and see the lateral sulcus here separating the temporal lobe down below from the parietal lobe at the top. So let's just do a quick recap. So in this lecture, we basically talked about the 3D anatomy of the basal nuclei. We highlighted some of the important functions of the basal nuclei as well. We contextualized the location of the basal nuclei in the overall hierarchy of the motor system organization. And then we talked about the location of the basal nuclei in the horizontal and the coronal slices taken through the brain. I hope you benefited from this video. If yes, then please do like the video and don't forget to subscribe to the channel. See you in the next video on the basal nuclei, where we will be talking about the neural circuitry of the basal nuclei in much more detail. Bye for now.